All right. Thanks for having me. Happy to be here. And now for something completely different, as they say. So Frank's exactly right. We don't necessarily think of rare diseases as having big data, at least as far as the number of samples. And I've spent a lot of my time in my research career kind of thinking about this question, or not exactly this question, but something like it. And as Frank mentioned, I'm at the Childhood Cancer Data Lab, which is an initiative of Alex's Lemonade Stand Foundation, which some of you may be familiar with. It's based in the area. And you can imagine, based on the name, that we are concerned with data and lots of it. And we work in the childhood cancer space. So what does this have to do with big data and rare diseases? How does that get connected? So all childhood cancer is in our mission, and childhood cancer isn't one disease. There are 12 major subtypes, some of which are quite rare. And even in the case of more common diseases, there can be significant heterogeneity, and I think we talked a bit about that today already. So there are 12 major types, over 100 subtypes, right? So even common diseases are made up kind of rare manifestations or conditions, and they're complex. So if you have a rare disease, there's only so many patients who get that condition. That makes sense. And only some of them will be part of research. So I've spent a lot of time. So before I worked in the childhood cancer space, I worked on rare autoimmune diseases. And I spent a lot of time thinking about this question. So what do I do if my data can only get so big? So I'm a transcriptomics person, so we measure, I mostly work in human, thousands of genes, so things get really long, but not very wide, so I don't have a lot of samples, and that can be trouble when we're talking about machine learning, as I'm sure many practitioners in the room know. So what kind of data am I talking about? So I work in with transcriptome data, and there's a good reason to focus on that first if you're going to be part of kind of a data lab. And the reason is there's a ton of it. So there's lots. It's grown over time. There's a really strong culture of sharing in this community. So over the past, over 15 years at this point, we've seen a growth in the number of samples or assays available. And this is just one repository that you can get from. If you look at all the publicly available data, there are probably about 3.8 million assays that are available. And now, this is not limited to human, this is a lot broader. And if you think about what these assays typically go for when you take into account reagents and such, it's about $1,000 an assay, and that's about $3.8 billion that we've already invested in data of this type. And it's downloadable now. So these are all publicly available. So you can go process it if you'd like. Obviously, that's challenging. The devs tell me this is, in fact, petabyte scale, and I'm inclined to believe them. So just to make sure everybody's on the same page, what do I mean when I say transcriptome data? I'm talking about the collection of RNA molecules in a sample, genome-wide. So let's, again, make sure everybody's on the same page. Go back to the central dogma, which you may or may not have seen back in high school biology class. So if we start with DNA, your genome, everything that could be coded and turned into a protein. I'm talking about RNA. So RNA is then this kind of intermediate step that's transcribed and then translated into proteins, right? So the actual building blocks of life. And then those come together to form cells and tissues and ultimately you. So Rich Jones, one of our software engineers, likes this analogy. You go from your hard drive to your RAM, which is analogous to your RNA here, CPU to your program. I am obsessed with Great British Bake Off. So I like this analogy better. So if a cookbook is a set of everything that you could possibly make, so that's your DNA, then the recipe is the part that you've taken out to actually go ahead and make. Then you make your product, which is your cupcake, any kind of baked goods. And then a collection of baked goods then turns into a bake shop, right? So that's everything in a bake shop. So just to reiterate, cookbook, DNA, everything that you could possibly make, recipe, RNA, what you're gonna make, cupcake, your building block, and then you get your bake shop, which can have all different kinds of baked goods in it. A wedding cake, some gingerbread cookies. It's fall. It's good to think about. So when I say transcriptome, I'm talking about all the recipes. So that's to say, if I have all the RNA in the sample, say, imagine every time I have a recipe, I can infer that I'm gonna make a cupcake. So however many copies of the cupcake recipe I have, that tells me about how many cupcakes might be in the bake shop, right? So the idea being, I try to infer the bake shop. So if I've measured all the recipes, maybe I can find out what's going on. That's the idea with transcriptome data. Given the collection of RNA molecules in the cell, what is happening in that tissue at the time of biopsy? So something like a tumor sample, what cell types are there? What's going on? Now, whether or not that's actually what you want to measure for debate, and Michael makes some really excellent points about, sampling time is really important. So that transcriptome data, thanks for following me on that cake-based digression. It's out there. So why can't people use it? So why don't people use this resource, or it's really challenging to use? And the reason is people have designed, it's a collection of individual experiments, right? People have designed their experiments to do a specific thing, to answer their question, and they process it accordingly. And the molecule that we're analyzing is inherently noisy. This data is poorly labeled. It's not clear how it's processed, so that is a problem as far as trust. So that makes it kind of difficult to use. So one of the first projects that we're doing, and it's in beta right now, it's called refine.bio, and that's the domain. And what it is this shovel-ready collection of gene expression data that you can go use. And we're trying to do it in a manner that's a little more transparent as far as our processing and such. So the idea is really simple. I start out with this. This is what lives in the publicly available kind of repositories. I want to uniformly process it so it looks like this, and it's obvious to you how it's been processed. And then I want to aggregate and normalize it. So go from small individual experiments to quote unquote big data. So I want to talk about, I think, kind of both parts of this are valuable for the kinds of things that we're talking about today, which is AI and precision medicine. So in the first example, say I'm just looking at kind of these uniformly processed data, how can that help me? So if I'm a researcher and I'm kind of doing an investigation, looking at transcriptome data, I have a cohort of interest. It's small, maybe I'm working on a rare disease, and maybe I'm studying treatment. So if I have baseline biopsies, and I know that I have a group of patients that responds to treatment, and unfortunately, I also have a group that does not respond to treatment. So I can, train a supervised model. Maybe I can draw this line really well in my own data, or overfit it, perhaps. But if I have access to uniformly processed data, I can search for my disease and treatment combination. And maybe it turns out 6 other groups out there have done something similar-ish to what I'm after. So what does that help me do? Right? Because all models are wrong, some models are useful. We want to check that our model is useful. And it's only useful if it's generalizable, as we talked a little bit about today already. So what I could do if I've identified these samples, maybe I can ask if this particular model works in girls or older kids or kids from different genetic backgrounds, right? Because my experiment is determined, there's collection bias, right? It depends on who I see at my center, for example. And other centers might have a different group of patients. So having access to these uniformly processed data could be really helpful. This is kind of an example that's maybe closer to translational, but this is really very useful for basic biology as well. And I think that's could be very powerful. So that's this half. What about the aggregation and normalization part? So what about putting everything together? How can that help us? So I don't know how many people are familiar with gene expression data, but it's highly structured, right? So genes tend to vary together, tend to function in similar biological processes. We've shown that over and over again. one of the early techniques to kind of suss that out, to get these patterns out, is called co-expression module analysis, and I'm going to call that a modular framework. And I worked on this for a while as a graduate student, but the idea is this. I have my individual data set. I want to see what genes are co-expressed, what likely function together in my individual data set, and I get sets of genes that are lumped together on the basis of their expression pattern. And that is pretty useful because measuring individual genes, especially if you're going to try and make that generalizable, is a little tricky, and that's fine. So you could do that. Maybe I have 4 data sets, or like the example above, but then that becomes a problem with scale, right? There's no automated way to say this module is the same module I saw over here. There are ways, but it does require a lot of human intervention. So this doesn't scale very well. So when I was a postdoc at Penn just a few months ago, I thought about how we might sidestep this problem, right? So how can we learn patterns that we can then possibly transfer to data sets of interest so that we're using the same model to describe them? And what does kind of having big data do for us? So in the transcription phase, we have a gene by sample matrix that's often pretty noisy. If we go at what we want is this sample by pattern, so a co-expression module or a latent variable component of some kind, which are these combinations of genes, which again are thought to be and are often have been shown to be more robust than measuring individual genes. And once I've learned these patterns, I can apply them to my new data set and ask questions like, is this pattern higher in disease versus controls? So that is what I set out to do. Again, to revisit our modular framework, I could do it this way. Then I have 4 different models that I have to compare across. Or I can use aggregated normalized data. So I'm going to use a compendium called Recount2, which is from Jeff Leake's group at Hopkins. And what I can do is train a model. The type of model is called PLIER, which is from Maria Cecchina's group at Pitt. And what I can do is then this particular data set turns out to be what I ended up using is 35,000 human samples. So that's a lot of different biological conditions, a lot of diseases. And I can learn how genes tend to vary with each other. So it go from this problem, I don't know how to kind of look across, to this. I learn, I transfer my model, and then my patterns are the same. So we have a preprint on this. This is just case sensitive, just a heads up. If you want to learn more, I'm not going to go too much into the details here. But I studied, while I was at Penn, ANCA-associated vasculitis, which is a collection of rare autoimmune diseases. And Recount2 contains no vasculitis data as far as to my knowledge. So if we train on Recount2, we have this model we call MultiPLIER. We kind of project our ANCA-associated vasculitis data into that space. We see we have groups of patients, those with more severe disease, those with less severe disease, and controls. And we can see that there are differential expression of this particular LV. And then we can ask what genes kind of go together to in this pattern. And if we look, the top ranked genes are things that folks who have this particular disease raise antibodies to. So even though we haven't seen any vasculitis data, because biology is shared, and this is something normal about biology that is kind of aberrant in disease, we can pick that up as being fundamental. So I don't want to go into too much detail here, but it kind of is intuitive that if we're learning across a bunch of different conditions, that we're going to capture more biological signal than if we look at a single tissue or single disorder. So this is a comparison to lupus whole blood data, and we can see that we get more patterns out when we train on the full Recount2 data set, more pathways, so biological functions are captured, and we think that that could be really useful for studying rare diseases. And look forward that could help us here. So if we train a PLIER model on our single data set, because it's this kind of data intensive matrix factorization approach, kind of signals kind of get lumped in with one another. So I have a relatively few patterns or features, and maybe if I have truly 3 groups of patients that would benefit from being treated differently, if I just look at the features from my individual data set, I'm not able to suss that out. But if I have a model that has more features and are better able to separate signals just by the breadth of different conditions, perhaps I'm able then to kind of more easily separate these. So this is an idea of where kind of this large body of transcription data can help us. So I just want to close with some quotes that stick with me anytime I think about this kind of problem. So this is Daniel Himmelstein, who is a postdoc with me at Penn. He says, on big data, data collection biases are always larger than statistical uncertainty. So when I think about what we work on with the transcriptome data, different cohorts, right? Biomedical research for a long time focused a lot on men, for example. So there are collection biases all in everything that we have. And as a kind of result of these collection biases, Casey Green, who's the director of the CCDL, likes to say, now domain expertise matters more, not less. So that's to say, I could have fit my model, I could have applied it to ANCA-associated vasculitis data, and perhaps, I was looking at a trial. If I'm not familiar with the area, and I would argue that I'm not that familiar, I might not know that patients stay kind of in trials if they tend to feel better, right? There is some kind of bias. And if I am just a practitioner and I don't have a team around me that can help me interpret my data, that can be problematic or challenging. And that's all I wanted to leave you with.